Hello, everyone. My name is Hamed Eftiari, and I'm delighted to be in this symposium in CPDD 2022. So we are going to talk about optimization of what we call transcranial electrical stimulation. I do not have any conflict of interest. Before starting my talk, I should say that I need to thank my colleagues and collaborators here in University of Minnesota. I came here less than a year ago, and I'm really delighted to find a network of colleagues and collaborators and supervisors and mentors who help me to be able to move forward with the research I'm doing. So what we have in drug addiction, we are interested to find networks and circuits that are involved in addictive behaviors. And we try to target those networks and circuits with the hope to modulate what we have in addictive behaviors. And to be able to do that, we have different non-invasive brain stimulation technologies. We have focused ultrasound, transcranial magnetic stimulation, and electrical stimulation. And Colin Hanlon is going to talk about TMS, and I'm going to talk about what we call transcranial electrical stimulation. With TES, we have different modalities. We have direct current, alternating current, random noise. So there are different modalities. And what it actually looks in my lab, it's just what we use as high definition, multi-site, alternating or direct current stimulation. And this is how we target different brain networks. The promise of using electrical stimulation is we probably can have portable devices that could be used easily in the moments that people need to have kind of some modulation in their brain activities, while it would be hard to have them in clinics from like 5 a.m. when they have kind of issues with drug craving. So that is the promise of scalability for electrical stimulation. And what we do with transcranial electrical stimulation, we welcome other interventions. We are interested to see how we might be able to modulate drug craving. And we do that with different drugs. This is the study that we have done with MET users, stimulating dorsolateral prefrontal cortex, trying to see how we might be able to show there is a significant reduction in craving when we expose people to brain stimulation. And there are different studies. People have done different studies in terms of how electrical stimulation and magnetic stimulation could modulate drug craving. And there are variations in response. This is a new trial that we have recently published in Human Brain Mapping. It is not still there, but it has been accepted to be published in Human Brain Mapping. But what we can see here, there is a significant variation in response to electrical stimulation, and you can see that we have significant variation in terms of the amount of electrical current that these 60 patients will receive in their brain when we do the electrical stimulation. So how we might be able to consider these individual variations and bring in biomarkers that we have, brain-based biomarkers to inform optimized stimulation. To be able to do that, we have to consider 4 levels of individualization: context, target, dose, and timing. And during my talk, I'm going to address these 4 major levels of individualization. For context, to be able to consider interindividual variations in terms of the type of drugs that they use, the context of drug use, the situation that they have drug craving, what we call drug cue reactivity. So we provide large databases of drug-related situations, and that would give people this opportunity to be able to get to the context of drug use. And there are inter-individual variations. I think that Colin is going to talk about that level of priming for making people ready for the stimulation. And that's another interesting topic for discussion. I'm not going to go to the details about that, but it might be interesting for you that we have recently made a consortium of labs that are interested to extend fMRI drug cue reactivity, and we collected something like 2,000 participants, data that are being collected from 2,000 participants for fMRI drug cue reactivity, and we are gonna extend that international collaboration. And if you have any sort of fMRI drug cue reactivity data, I would highly invite you to that consortium, and that would be a great opportunity for collaboration and kind of bringing that contextual information to what we are interested to. The second level is what we call target. So what would be the areas that we are interested to target? And there are many variations in terms of the targets that we can do, and then how we consider inter-individual variations in targeting. One of the targets that we are interested to is what we call frontoparietal network. And we know frontoparietal network is involved in executive control, and we know that that would be an important part of what we call drug craving control. In a recent study among 60 met users, we tried to see if we stimulate the classic frontoparietal kind of nodes, F3, P3, F4, P4. Can we engage frontoparietal connectivity? But as you can see here, there are kind of interaction between right, left, F3, F4, or P3, P4, but there is no frontoparietal engagement, as you can see here in this figure. But the question is, do we have any frontoparietal engagement in drug cure activity? So we use seeds in the right and left, F3, F4, and we try to see if there is any kind of frontoparietal engagement. And this is a whole brain analysis. And you can see that, yes, there are parietal nodes that are involved in interacting with frontal node during drug cure activity, but they are not actually right under what we call P3, P4. If we do modeling and try to see what are those areas and how we might be able to target those areas, you can see those areas are located under CP5 and CP6. So we probably need to change the montage to be able to target those networks that are involved in drug cure activity. And if we change those targets, you can see that we can have significant level of engagement of frontoparietal connectivity, which makes it even more interesting. But one of the challenges that we have, as you can see here, I mean, people are different in terms of the variation. As you can see here, people can have different nodes of, or the peak maximum electricity in these areas of the brain. This is the frontal side. And that would make a question in terms of should we consider those interindividual variations within the frontoparietal network, the average frontoparietal network that we found to be targeting. The third level is about dose, how much electricity that we should consider in stimulation. If we do classic montage for frontoparietal synchronization, I mean F3, P3, F4, P4, you can see here that if we deliver something like 2 milliamp in each side, we can have a variation in response among individuals between something like 0.1 to 0.4. So this is a kind of a variation, and this is a histogram of distribution of peak electrical dose that we give people to those nodes, which is significant. So you need to think about, okay, how we can consider those interindividual variations. And then if we go for the informed montage that we are discussing about, something like F3, CP5, F4, CP6, then things would be even more complex, because in the parietal side, we have a higher level of variations, and then there is no, even in the average, they are not equal in terms of the level of electricity they receive. And we need to basically change the amount of electrical currents that we deliver to each node to be able to at least have a balance between nodes. But we still have interindividual variation in terms of the level of electrical current that they receive, which is another important factor to be considered. The fourth factor is about timing. And then we talk about timing. There are multiple levels of timing that is important for individualization. One of those levels is what we do in the frontoparietal synchronization, because for frontoparietal synchronization, we use transcranial alternating current stimulation. So we use two nodes that are going to be synchronized with the same frequency, and that is the way that we get them connected in alternating dual side alternating current stimulation. So we have a large sort of frequency phase. So we can go from 6 hertz to 30 hertz. So there is a variation in terms of what would be the best oscillator rate to engage frontoparietal network. And it's not only about the frequency, it's also about the phase differences between two nodes of a stimulation. Should we do the alternating current stimulation in one node ahead of the other? So there would be a phase difference between electrical stimulation in both sides. And the question is, what is the right target for being able to do that? As you can imagine, there is a large parameter space for that in terms of what would be the right frequency and phase in each individual level. And there are variations in what will be the best target for each individual. To be able to address this, which is a bit complex problem to solve. What we are trying to do is putting people inside the scanner and do the stimulation inside the scanner and optimize stimulation for each individual. So we are interested to have a specific target, frontoparietal target, and engage that target. And we are interested to optimize that engagement and optimize the stimulation parameters based on that target engagement. This is what we call closed loop TSFMR. So trying to make a closed loop of stimulation inside the scanner. And to be able to do that, we usually start the stimulation with watermelons, and you can see that you can actually see inside the watermelon with MRI, as you can imagine. And then, of course, we eat watermelons after stimulation. And after doing the kind of the initial technical scans with watermelon, the next subject that should go inside the scan is the PI of the project. So I've done several sessions of electrocardiogram stimulation inside the scanner, and there are kind of technical issues that we have been able to put those things together with a postdoc and a PhD student that we had in my lab in the University of Oklahoma. And now you can see we have put together a checklist, technical checklist of how to do concurrent TSFMR inside the scanner. And going beyond that in terms of some technical issues, because in this put the subject inside the scanner, we need to make sure there is no electrode on the back of the head. There are some modifications that we had to do in the electrodes and some innovations for being able to do that. We put together that kind of technical details in a technical note that is right now in bioRxiv, but it's going to be published in Brain Bank Behavior. So all those technical details are there. And we recently have done a small trial with working memory engagement in healthy participants, trying to see if we can engage the frontoparietal network. Recruited 10 participants in each group, and I'm not going to talk about the details about that, but the only kind of thing I'm going to talk is just, as you can see here in both groups, the level of side effects in both groups are I mean, there's no significant difference between the level of side effects that we have in each group, which is promising. So it seems that technically this is doable. And we made a group of people who are interested to do electrical stimulation inside the scanner. And now we have a consortium of labs that are doing the same. And if you are interested, that is going to be something interesting to do. And you are welcome to join our consortium. Some take-home notes. So we discussed about transcranial electrical stimulation, especially when they are portable and digitized, there are definitely potentials. We know that right now we have 67 papers, original trials with electrical stimulation, 108 papers with RTMS, large intellectual variability that we have in this field. We try to use structural MRI and functional MRI to inform the parameter space in both group level and individual level. We discussed that concurrent TSFMR is feasible. That's an interesting technology. We developed a checklist for that contest checklist. We try to close the loop, and it seems that the closing the loop is technically feasible, but there's still devils in the details. So there are still some technical challenges for that. And what would be the target that we are interested to engage? We discussed about frontoparietal network, but there are still lots of interesting discussions about other targets. So if you are interested, we have a webinar with Colin and other colleagues discussing about that on July 27th. So you are welcome to join us. You can find my email. You can send me an email if you're interested to. And I should thank my colleagues and collaborators and those who helped me to make this happen. Thank you very much for your attention.